Hello and welcome to my YouTube channel Lighting the Knowledge Candle. In this video, we will be discussing about the clinical therapeutic trials. Clinical therapeutic trial is an experimental study involving humans. It compares the therapeutic benefits of greater than or equal to two interventions. Example, treatment versus placebo or treatment versus treatment. In order to improve the quality of clinical therapeutic trials, it is randomized, controlled, and double blinded. Neither the subject nor the researcher knows when the subject is in whether the subject is in treatment group or control group. This is known as double blinding. Additional blinding of the researcher analyzing the data is known as triple blinding. We are going to study four types of clinical trials here. Crossover clinical trial, intention to treat analysis, as treated analysis, per protocol analysis. Crossover clinical trial actually means it compares the effect of a series of greater than or equal to two treatments on a subject. The order in which subject receives the treatment is randomized. Key points to be remembered here in crossover clinical trial are washout period occurs between the treatments, and here subjects serve as their own controls. Once again, I would like to repeat the crossover clinical trial. Crossover clinical trial compares the effect of a series of greater than or equal to two treatments on a subject. The order in which subject receives the treatment is randomized. Key points to be remembered here are washout period occurs in between the treatments and subjects serve as their own controls. Intention to treat analysis. In intention to treat analysis, all subjects are analyzed according to their original randomly assigned treatment. No one is excluded. Here, attempts are made to avoid bias from attrition, crossover, loss to study, non random compliance, but may dilute the true effects of intervention. As treated analysis, all subjects are analyzed according to the treatment they actually received. So there would be increased risk of bias in as treated analysis. Per protocol analysis, subjects who fail to complete treatment as originally randomly assigned are excluded. We can also see increased risk of bias here in per protocol analysis. Then preclinical studies will be followed by 5 phases of clinical trials, which can be remembered with the mnemonic. The components of these 5 phases can be remembered with the mnemonic Can I Swim? As we all know, phases of clinical trials is an important topic for our exam. First, in preclinical studies, drug is tested in lab animals and in vitro experiments. In the preclinical studies, next phase is phase 0. Can I swim? The mnemonic starts with I swim. Initial pharmacokinetic and pharmacodynamic assessment will be done via microdosing. Phase 0 is often skipped. In phase 0, there would be sample size which is very small and the number of healthy volunteers or patients with disease of interest. With the small number of healthy volunteers or patients with the disease of interest. It is open label. Next, phase 1. Safety assessment via dose escalation. Safety assessment. Can I swim? S. Safety assessment. S for safety assessment. Safety assessment via dose escalation. Determine maximum tolerable dose. Maximum tolerable dose could be known in phase 1 of clinical trials. This could be an MCQ. Small number of healthy volunteers or patients with a disease of interest. This is also open label. Next, phase 2. Swim. W. Does it work? Efficacy assessment. Efficacy assessment could be assessed in phase 2. Provides additional data on short-term adverse effect. The sample size here would be moderate number of patients with disease of interest, randomized, controlled, anonymized. Phase 3. Effectiveness assessment. Phase 2 would be efficacy assessment. Phase 3 would be effectiveness assessment via comparison with current standard of care or placebo. I. Swim. SWI. Any improvement. I. Improvement. Here the sample size would be large number of patients with disease of interest, randomized, controlled, anonymized. moderate number in phase 2, large number in phase 3. Phase 4 provides data on long term or rare adverse effects. Can it stay on the market? Can it stay on the market? Post marketing surveillance of patients with the disease of onset is done in phase 4. This is all about the clinical trials discussed in first aid.